We're gonna go ahead and get started. Thank you, everyone, for joining us. It's great to see this auditorium getting full, and hopefully we'll probably have some late stragglers, but it is a minute after 5:30, so we'll go ahead and get started. And thank you to our Zoom webinar listeners and joiners. Thank you for joining us online as well. Really excited to welcome everybody to our 68th annual research lecture. Now longstanding tradition here at the University of New Mexico, and one of the highest honors that is bestowed on faculty here at the University of New Mexico. To get us started, of course, it never fails when I'm in this room. I have to pause. I'd like to recognize, founded in 1889, the University of New Mexico sits on the traditional homelands of the Pueblo of Sandia. The original peoples of New Mexico, Pueblo, Navajo, and Apache, since time immemorial, have deep connections to the land and have made significant contributions to the broader community statewide. We honor the land itself and those who remain stewards of this land throughout the generations, and also acknowledge our committed relationship to Indigenous peoples. We gratefully recognize our history. It is now my pleasure to welcome Vice President for Research, Ellen Fisher. Sorry.
I'm so happy to be here. Melissa Thompson was going to do the introductory remarks because I literally just flew in from Dallas, and boy, are my arms tired. I have to take just a second and say that I was at the Hispanic Serving Research Universities, HSRU Alliance Summit meeting, which is very interesting. For those of you who don't know, there were 21, and as of this meeting, there are now 22 Hispanic Serving Research One R1 institutions across the nation. It was very interesting to be there and to think about the unique aspects of institutions that are both Hispanic serving and Research One, very high activity research institutions. And as I was thinking about that and what that means, and then thinking about the history of UNM in general, it turns out that that designation just went into effect in the 90s. But prior to that, since the beginning, UNM has been a Hispanic serving institution. And I just wanted to recognize that after hearing the land acknowledgement and being 2 days at this conference where we were all talking about the fact that we have that designation from the US government, but even prior to it being a designation recognized by the government, this institution was already a Hispanic serving institution. And we've also had the designation of being a high activity research institution for many, many decades. And so with that, I want to just say that it's my great pleasure to be back here in time to do this. In fact, I had my flights arranged so that I would be back in time to be at this lecture, because it is one of the most important things that we do, where we recognize the outstanding, absolutely amazing research that happens here at the University of New Mexico. It's amazing to me to think about the fact that Felisa is the 68th recipient of this award at UNM. So this is a tradition that goes back, I get to say I don't get to say this very often. It dates back to before I was born. Okay. These days, there's not that many things that have lasted that long. So that's kind of nice. And I think that while we have other ways of acknowledging the great work that's being done on this campus, I think this is one way that really helps us put a spotlight on the specific achievements of one of our distinguished faculty. I would say that this is interesting to see how Professor Smith's work complements and elevates the legacy. I don't know how many times we've had show and tell in those 68 years. I'm so fascinated that we have show and tell. I'm excited to hear more about that. Dr. Smith is a mammologist focusing her research interests on paleoecological and evolutionary research. And her talk today, which I'm not going to introduce her right now, but her talk today is also the title of her latest book, mammalian paleoecology, using the past to study the present. And that again, sort of harkens back for me, having been at this conference and thinking about the history of these institutions, and the place and what we learn from looking at our past, and what that tells us about our future. I will just say that one of the things I was asked to do when we announced this award, and I was asked to do have a quote for the press release. I'll just tell you what I said, that Dr. Smith's work and dedication represents a powerful contribution to the field of evolutionary biology by focusing on the lives of ancient mammals. This field provides a unique insight into both the process of evolution and how it has generated biodiversity across the globe. It also has enormous potential, and this is the key thing here, I think, to inform our collective future in critical areas such as sustainable agriculture, resilience to climate change, and genomic models for disease. So I'm really excited to hear the lecture. And so to do so, we have invited a distinguished professor emeritus, Dr. James Brown, to come and actually do the formal introduction of our speaker. I'm really excited. Jim, okay. Jim was a faculty member at UNM for more than 20 years. He's known as a pioneer in many fields, including population and community ecology and physiology. He also developed the field of macroecology and the metabolic theory of ecology. This is the really cool part. He was the 1999 recipient of the ARL. So it's a legacy here, which is very cool. And I think because of that, he was elected as a member of the National Academy of Sciences 6 years later. So with that, I will turn it over to Dr. Brown.
Okay, is this going to be on? Is it on now? Great. Well, thank you, and it's great to be back. My wife, Astrid Kovic Brown, who's sitting here, and I spent 25 wonderful years at UNM, and they were wonderful, both professionally rewarding and personally rewarding. And we'd undoubtedly still be here in Albuquerque, except that it doesn't have an ocean. So it's an honor and a pleasure to introduce Felisa Smith as the 68th annual research lecturer. This is one of the highest honors, as already been said, that UNM bestows on its faculty. And I'm going to mostly let her talk about her research. I thought I'd briefly mention some of her other contributions, professional contributions to UNM and to science. So she came here in 1992 as a postdoctoral researcher. She's gone up through the ranks, became full professor in 2011, and was awarded a distinguished professorship in 2023. We generally tend to evaluate our faculty members on 3 criteria: teaching, service, and research. And Felisa has excelled in all of those 3 areas. In teaching, she has taught many, too many for me to count, undergraduate and graduate courses. She's also mentored 7 postdoctoral researchers, 9 PhD students, 3 master's students, more than 30 undergraduate honors and other undergraduate students, and 1 high school student. She's also, sort of under the category of teaching, been amazingly involved in outreach activities, both presentations to the media, interviews, and in popular writings. Under the category of service, university and professional service, she has served as the president of the American Society of Mammalogists and the International Biogeography Society, so 2 international societies. And she served on countless committees, both for professional organizations and at UNM, on NSF grant review panels and so forth. Her research, which is what we're here for today, is also outstanding. Some of it's already been mentioned. More than 100 peer-reviewed scientific papers, 3 books, on the order of $4 million worth of funding, mostly from the National Science Foundation, and a major role in leading interdisciplinary interdisciplinary biological science and training in that. Her research, again, has already been mentioned, has been characterized, well, I mean, it's almost too diverse to characterize with a few words. She's worked on evolution of body size, especially in relation to climate change, the extinction of the Pleistocene megafauna, these giant mammals that were here up until just a few thousand years ago, and the role of humans in those extinctions and the role of those mammals in transforming their landscape, and then the effect of that extinction and the changes in the landscape and the other animals after they became extinct. She's also published papers and done considerable research on the role of women in science. And on top of all that, she's just an amazingly well-rounded, wonderful person, right? Her family is here. Her husband, Scott Elliott, who's recently retired from Lannel and is now teaching in public schools in Santa Fe. Her 2 daughters, Emma and Rosemary, one of whom has a PhD already and the other is going to get one in just a few months. And in addition to everything else, I think, she's just been a great person, an outstanding role model. She's a really dedicated feminist, very sort of tough, but also very gentle way. And let's hear about the research.
You gotta give me this finger, I can't talk.
It's yours now.
You all good? Yep, and everybody can hear okay? All right.
Oops, we good?
Yep. All right. Well, thank you very much. I want to start actually by just acknowledging how honored I am by this award. And I'm especially grateful to my longtime friend, mentor, and collaborator, Jim, for coming to introduce me to this lecture. Jim was my postdoc mentor all those years ago, and he's a person who has always believed in me and encouraged me in my wildest ideas. If you've ever heard him, his wildest ideas, that mine are kind of tamed by his wildest ideas. I'd also like to thank my wonderful colleagues in biology and elsewhere, to my students and postdocs and UNM actually, for providing an environment where you can do interdisciplinary science and you can do it well. And I think that kind of freedom to sort of go where your ideas take you is kind of unusual in academic settings. So I really appreciate that. I would also like, and I'll try not to get teary, to dedicate this talk to my mom, who passed away a few years ago, and who there's not a day where I don't think about her and miss her. Okay, so fundamentally, as you probably sort of heard, I'm a paleoecologist. That's what I call myself these days anyway, which means I'm interested in the ecology of long dead things, right? And really, most of that work is in the area of what we call conservation paleoecology, which is trying to use the past to understand current environmental problems. And hence, I've got this really spiffy lab motto that I stole from William Shakespeare's play, The Tempest. But you might say, why the past? Why is there a focus on the past? And to explain that, I would challenge you to consider that the average ecological study lasts 1 to 5 years, right? This actually hasn't changed in more than 25 or 30 years. This metric comes from this paper in 2021. I remember in 1989, back in the day, looking and it was about the same number, right? And why that number? Well, that's how long it takes a grad student to work on a degree, right? So time is not something you often get incorporated in ecology. So these studies produce a lot of super excellent, insightful findings. I'm not panning, modern ecology, but I would argue that there are a lot of pressing environmental issues that actually need data spanning a much longer time frame because they have indirect and direct effects that take longer. A tree lives for hundreds of years. So looking at the effects of losing trees can actually take a really long time. And so Dave Tillman, I put this quote up here, which I think is really informative. Ecologists, you can have a problem if ecologists use short-term experiments to try to address some of these long time issues. Because of this, for this reason, some 45 years ago, NSF saw the light and they established the LTR system, the long-term ecological network. We have one here, the SEV, you probably heard of the Sevietta. It's our LTR that's located south of Albuquerque. And these sites are actually set up to address this issue of collecting and compiling long-term ecological data, right? So they standardize the kind of data they collect. There's a series of them, and they've actually led to a lot of really powerful insights in how ecosystems work, which is important. But I would argue that there is long-term, and then there is long-term. And it turns out there are other records that span much longer timeframes, right? So we're talking about studies here. If you incorporate historical records or indigenous knowledge, you can push your time frame out, but the paleo record can actually allow you to incorporate really long timeframes. And that can be really helpful if you're trying to understand a process that takes decades, centuries, or millennia to play out, right? And let me give you an example of that. So let's say you have some environmental parameter. Let's say it's how animals respond to global warming or ocean warming, right? And you might have some data on ocean warming spanning a decade or so. And maybe this biotic response is something like whether recruitment or extinction rates are a function of temperature warming, right? But the problem is that you develop this predictive model, you make a suggestion, oh, this is what's going to happen if the oceans continue to warm. We need to worry about these plants because apparently you care about plants. Corey isn't here, so I can make comments like that. But if you actually incorporate the paleo record, you might find out that this is a nonlinear response and that your predictive model would be wrong because maybe pH matters too. And pH doesn't change over the time frame of a decade or whatnot, right? So this is, I think, why you can argue that paleo records can be useful. And the pressing question that I'm talking about then is biodiversity loss, right? I think we can all agree that this hand in hand with climate change is one of the most pressing environmental issues we have to deal with today. So what I want to do today then is talk about how the paleo record can inform questions that are relevant to biodiversity loss. So I'm not going to talk about a lot of the other work I've done. I'm going to just focus on this for this particular talk. And I'm going to try to kind of weave a narrative and tell you a story about this. And the story is gonna have two parts. The first part is I'm gonna address what the paleo record tells us about what characteristics predispose animals to extinction, right? And then the second part is I'm gonna talk about what the fossil record shows us about the consequences of biodiversity loss on ecosystems, right? And I am also, frankly, gonna skip most of the details. It's the evening, we get to have wine after this. So we're gonna just sort of talk through a lot of these processes. I'm gonna assume that most of you don't spend a lot of time thinking about Earth history. So let me give you the quick 20,000 foot view, short 1 minute view of Earth history, right? So here we have Earth history. This is a picture by Raymond Toll, who's a paleo artist in Homer, Alaska. He just does these great images and lets people use them, which is nice. It's a little misleading, however, because these are not to scale. So, you know, there's a different amount of time occurring in each of these. And then the other thing you need to realize is that this little bit here, this is 7/8 of all Earth history that's right there, that we're just sort of ignoring, right? So what we're gonna focus on really is the last 540 million years of a 4.6 billion year Earth history. So life starts. That was pretty important. That's about 3.6-ish billion years ago, give or take a couple 100 million. And then we're gonna skip all the inverts and all the other things because I am fundamentally a vertebrate ecologist. And we're gonna go straight to amphibians, which evolve in the Paleozoic, in the Devonian, Mississippian timeframe. Then we have reptiles, and then everybody's, unless you're a mammologist, everybody's favorite group, the dinosaurs, right? The non-avian dinosaurs, because the avians, the birds evolve a little later. And then we have mammals. Yay. This is about 210 million years ago, give or take about 10 million years. And then, oh goodness, birds evolve a little after mammals, I should point out. And then a really awful tragedy happens. By the way, there are about 5 mass extinctions here. I'm not talking about any of those, and they're not even really indicated on here. But there is a tragic event, and that is the non-avian dinosaurs meet their demise because the Earth gets smashed, right? Okay, so that turns out, this event, to be really important for mammal evolution, right? We evolve, because we are mammals, around 210 million years ago. We do nothing for a really long time, right? We don't get bigger than about 15 kilos during the Mesozoic. But after this extinction, mammals go nuts, right? And so there's all kinds of super interesting things that happen during this time period, which is known as the Cenozoic or the recent, right? The age of mammals, right? So we're going to talk about this time period a little bit, but we're also going to talk about this time period where super cool stuff that's pertinent to biodiversity loss happens. Okay. So now I'm sure you're all experts on Earth history, and I can use buzzwords and you'll understand them all, I hope. Okay, so the first question I want to address is the characteristics that could predispose mammals to extinction, right? And to do this, the time frame I'm using is the entire Cenozoic, where we're going to take time here and we're going to bin time in 1 million year intervals. And we're going to collect data, or we have collected data because this has been done, on extinctions, right? What animals went extinct, which animals survived in those 1 million year bins. And then we're also going to talk about the last 125,000 years. That will use a slightly different data set. So just so you know that we have data, we use 2 different global compilations we've put together over the years. You don't really need to know much about them except for they're global compilations of mammals, right? And they have a lot of information, and it took a really long time to put them together. This one especially took us years. Jim was part of this group that put that one together. And so for each of these, we have also the duration of the species, right? Okay, so the first question you want to know is, what does mammal turnover look like? over 65 million years, right? And by turnover, what I mean is the change in species diversity. That is originations, new species evolving, speciation, minus extinctions. And this is plotted from when the dinosaurs went extinct, non-avian dinosaurs, to the modern, and this is turnover. And what you can see is it fluctuates. It fluctuates a lot, right? It fluctuates because the mean duration of a mammal taxa is about 2 million years. So species are going extinct all the time. We call that background extinctions, right? And new species are rising as well. And what's really kind of clear though, is there isn't a directional trend to turnover, right? So, but you might say, well, okay, that's true. But what about some of these factors that we think impact extinction in particular, not just turnover? What about temperature, for example? And we can get temperature records for the Cenozoic. So this is from Jim Zakos at UC Santa Cruz, where he looked at forams and looked at the Delta O18 and was able to derive paleo temperatures. So we've been to these data in 1 million year bins too. So this is the proxy. This is the APG, and then this is modern. So 10 million year bins. This is cooler, this is warmer. And what you can see is there is a lot of fluctuation in climate over the last 66 million years, right? So the question is extinction related to this, right? And so we took our extinction metrics and we plotted them against temperature. And the answer is no, right? So I'm going to show you the different ways that you could talk about temperature. This is mean temperature in one of these 1 million year bins versus the extinction rate in that bin. Notice some of these extinction rates are really high because this starts at 66 million years ago, and there's a lot of turnover at various points in the Cenozoic. So the mean temperature doesn't matter. The temperature variation between each of these million year bins, the abruptness of changes, doesn't influence it, nor does the variation within a bin. So temperature doesn't drive extinction in the mammal fossil record, right? Well, you know, we're all worried about climate change. Does this mean we have nothing to worry about? Okay, we're going to get back to that in a second. One thing I do want to mention though, because temperature is an important player, what it's important in is not extinction, but in evolution, right? So what climate does is it changes, it selects for larger or smaller body size of species, but it doesn't lead to extinction. And you can see this here where we plotted the maximum body mass of mammals on the different continents from about 100 million years ago to modern. And you can see there's this is where the KPG happens. There's a lot of fluctuations, they get bigger. The maximum body sizes that are achieved by mammals are achieved when it's cooler, right? And I'm not going to show you all the data or the analyses, so just assume that I'm right, I guess. So things like Indocytherium, for example, arose during the Oligocene of Eurasia at a time period where the Earth was cooler, right? So it's not that temperature isn't important, it's that it doesn't drive extinction, it drives evolution, which is a different thing. Okay, so another question you might ask, all right, well, you're puzzled, right? Because what we hear today is that large animals are at risk of extinction, right? We're all worried about elephants and rhinos and things like this. So you might ask, is large body size a predictor for extinction risk? Do larger things tend to go extinct faster? And how would one measure that? How would you figure that out? And so I'm going to walk you through how you do this. You do this using this thing that looks complicated, and it kind of is, but it sort of isn't also. This is what we call logistic regression. And the way you read this, and I'm showing you this for trilobites, everyone's favorite animal of the Paleozoic. There are 15,000 different species of trilobites that existed at this time period, or back 500 million years ago in Earth history. Each of these values is a selectivity metric. And the way you read this is that if the value is above the 0 line, larger animals, in this case trilobites, are more prone to extinction. If the value is below the line, smaller animals are more prone to extinction. And if the value is on the line, there is no selectivity. That is, body size does not contribute to extinction risk, right? The important thing is that includes the bars, right? So do you see any bars, anything here where the values don't cross the line? No, right? What that tells you is for trilobites, over about 100 million year time period, body size never led to increased extinction risk, neither small or large, right? Okay, and these, I should mention, are orders of magnitude. This is a really powerful analysis that's commonly used. Okay, so I've walked you through for trilobites. Now I'm going to do something I probably shouldn't do at 5:30 in the evening, and I'm going to show you a whole bunch of data. So here are the trilobites down here. Here are dinoflagellates, diatoms, articulate brachiopods, bivalves, clams, and gastropods or snails, right? And these span different time periods, but you can see we're talking about 500 million years of evolutionary history, right? Is there a directional trend? Do any of these groups show size selectivity? And the answer is no, right? Body size is not important in terms of extinction risk in these organisms. Now, it's kind of a long story why that's not the case, and I'm just gonna not even talk about this today, but I'll just tell you the reason is that body size is correlated with larger geographic range. And geographic range is the one factor that reliably in the fossil record buffers against extinction risk. And it's really ubiquitous across all different taxa. So the fact that large animals might have slower life histories doesn't matter because they have larger geographic ranges, right? Okay, so I showed you this for all these groups, but you may be saying, but they're not vertebrates. Mammals are special snowflakes, right? So are they? And we can ask the same question. But to do that, we have to estimate body size for mammals. So how does one do that? And this is something my lab likes to do, and that's what we do with all these things that are here. It turns out that you can estimate body mass for virtually any cranial or postcranial element. So for example, here, we could measure the length of this lower first molar. That's one of the most reliable predictors of body mass. We could also take this femur, we could do the overall length, we could do the distal or proximal head or the mid shaft width, any of these would work. Once we measure that, we use a predictive equation, which we spent a long time developing, right? So we developed these equations based on modern taxa, put these values in, and then that lets us estimate the body size of an animal. So this terminal Pleistocene gray wolf from Bering Sinkhole in Texas weighed about 36.5 kilograms or about 80 pounds, right? So that's how you get body size. And you can do that for tooth rows as well and other things. Okay, so you know how to do body size, you know how to read a selectivity graph. So let me ask, does body size influence extinction risk for mammals across 66 million years? And what a surprise, the answer is no, it does not, right? So again, here's 66 million years. Here's the selectivity, the log-SOD ratio you just looked at. Again, larger animals would be more prone, smaller animals would be more prone. The error bars overlap 0, it's not significant. Only a couple of these are significant, and there's clearly no directional trend, right, over the fossil record. So this is super interesting and probably again related to geographic range. So then you can say, well, you'll notice cleverly that I haven't filled out this last little blue bit, which is the last 125,000 years. So let's expand on that. And what we find is that over the last 120,000 years, it changes. Suddenly, large-bodied animals are highly susceptible to extinction risk. And these are 3 orders of magnitude. difference in extinction risk, right? So at various time periods, and we've even calculated what extinction risk is for large bodied mammals in the future if everything in an IUCN category of threatened or vulnerable actually goes extinct. So the pattern has changed, right? And moreover, it's interesting because if you break it down further, it's disproportionately higher for herbivores, these green squares, than say carnivores, these black squares. Here, instead of extinction risk, the log-odd ratio, I simply showed you the log difference between something that went extinct and something that didn't go extinct during one of these time intervals here, right? And you can see there's a 3 order of magnitude difference in the herbivores that go extinct, the size of the herbivores that go extinct in the late Pleistocene versus the ones that do not. So this is strongly and hugely size selective. Okay, so that's interesting. So what happened in the late Pleistocene or the late Quaternary that led to this? Well, it turns out that this pattern is correlated with the movement of hominids across the globe, right? And by hominids, I use the term hominids because it's not just humans, it's modern and archaic humans, it's also other species of Homo, right, that were present at the time. And you can see this here. Here's a map from Oppenheimer in 2012 showing the pattern of movement of the genus Homo across the globe. Some of these dates are argued about and they make some of them may be a little wrong, but in terms of our own species, Homo sapien, the exodus of Homo sapiens out of Africa into Eurasia started by at least 100,000 years ago in what were probably a couple of orbitally paced migration episodes. In other words, when climate shifted, there were more favorable corridors for them to migrate out of. And we spread across the globe pretty rapidly, reaching Australia by, at least 50 to 60,000 years ago, and the Americas by 14 or 15,000 years ago, maybe a little earlier. There's a lot of controversy and work on where people actually got and when they got there. And while I'm showing you this migration pattern for Homo sapiens, recall, as I mentioned, that other species of hominids were also present and coexisting in these ecosystems in one form or another till maybe as recently as 30,000 years ago, right? And we grew in number, right? So this figure comes from Timmerman and Friedrich a few years ago in Nature, and it goes, I think these are 5,000, I don't know. They're intervals, starting about 125,000 years, going to 1,000 years. And these are number of individuals, this is just Homo sapiens, per 100 square kilometers in little grid cells. And you can see the movement and then the piling up of peoples across the globe, right? So this is important because as people moved across the landscape, they changed things, right? Humans are rather novel and effective predators. So our movement across the globe had impacts. You can, whoops, sorry, right? And indeed, human migration was accompanied by a temporally and spatially transgressive extinction of mammals. And I'm going to show you what I mean by that here. So what I'm plotting is mean body mass on a continent. And all the major continents are here. Old World are shown in black, New World in blue, Australia, which is its own special place, in red. And you can see at the beginning of this time frame, most of the major continents had about the same mean body mass, right? With the exception of Africa, where body mass was already lower than it was on the other major continents. This is a little weird because body mass, the mean and maximum body mass of animals on a continent scales with the size of the continent, and Africa is the second largest continent. So you would have expected it to have larger animals, but it did not, which is kind of interesting. And you can see that for the other continents, Eurasia, North and South America, the mean body mass of mammals on the continent, and I should mention I'm excluding aquatic animals and volant mammals, bats. We're not talking about bats, right? terrestrial mammals was about 100 kilos, right? Very different than it is today. So when hominids got to Eurasia, what we see is suddenly there's a drop in body size that occurs when they enter that continent. And if you think about it, how do you get a drop in body size? Well, you get that because let's assume you have a mean distribution of mammal body sizes on a continent, and you have a given mean. You have selective extinction of one end of the body size distribution. What happens is the mean shifts down. And that's exactly what we see on each of these continents as humans step through them. So when humans get to Australia, there's an order of magnitude drop in the mean body size of mammals on that continent. Same thing occurs in the Americas as well. And it's interesting because these events occur when humans first enter a continent, but after a while, you don't see the same level of extinction. And I suspect that's because what you've done is you've lost all the naive prey, or excuse me, they were prey, but animal that hadn't seen humans before, right? And then some sort of stasis occurs. And in the future, if we lose all our vulnerable species, we'll actually end up with the average body size of animals on a continent being around 5 kilograms, right? That's really kind of sad when you consider it was 100 kilograms just 100,000 years ago. Moreover, this mammal extinction rate accelerated. So here's cumulative biodiversity loss over the last 150,000 years, while body size globally is declining. And this is all coincident with human population growth. All of these patterns are highly significant. And again, I told you I wasn't going to show you any of the data or correlations and stuff. So I'm not. You'll just have to accept that, well, you don't have to accept, you can challenge me. But anyway, that these patterns are real. Lastly, I would also point out that extinction intensity is related to body size. So this is the cumulative intensity of extinction over the last 120,000 years by different size classes. And you'll note that we've lost entirely several of the largest size classes on the planet. They don't exist anywhere anymore. And then the smaller ones, animals of 1 to 5 tons, are still, most of them are highly endangered or vulnerable, right? So interesting. So what has this done? What this has done is fundamentally reshaped the body size distribution of mammals on continents. And that's important because the distribution of mass of mammals on a continent reflects energy flow through each size class and ecological function. So this is just North and South America. Here are the extinct things. Here's extant, extinct, and extant. And you can see that the distribution without these things is fundamentally different, right? Okay, so why does this matter? It matters because, that was a rhetorical question, because the loss of ecological function can lead to changes in ecosystems, and really important changes. And a number of studies have shown this. This paper, this image comes from this paper by Jim Estes from UC Santa Cruz in Science of a decade ago. This paper transformed a lot of the science my lab was doing when it came out. And what you're seeing here is, here's an example of a cougar, a wolf, and a wildebeest. This is what the ecosystem looks like when they're not present. This is what the ecosystem looks like when they are present. This paper, in a very simple visual way, presented why these animals matter, right? And what happens when they're extirpated from an ecosystem. And it makes it clear that understanding what these changes are and what other changes will follow is really urgent. And because some of these changes are indirect and some are direct, they can take decades to play out. But waiting decades and decades for a long-term experiment to work before you try to mitigate is really at odds with the time-critical nature of extinction risk today. So this is why the fossil record can be really informative. So what we're going to do now then is move to the second part of my talk, which is on what the fossil record reveals about extinction risk. Okay, so again, here we are. Now we're gonna talk about this really interesting stuff in the Lake Quaternary. And what we're gonna do actually is we're gonna go local. So while we can, I could talk about this in multiple scales, including the global. Today, what I'm actually gonna focus on is work that we're doing in a mammal community in the Texas Hill Country on the Edwards Plateau. And it's near a town called Kerrville. And this is us, here's Texas, if you don't know where Texas is. Here's the Edwards Plateau. I know we try to pretend we don't know where Texas is, but it does exist here. The Texas Hill Country is about here, and this site is right about there. This is a really interesting cave site. You can sort of see it here. There, as far as I can tell, most of Texas is basically a bunch of fossils with a layer of limestone on top. And then every now and then a tractor or a truck falls in and they find another cave full of fossils. It is, the Edwards Plateau is the most phosphorus, I don't know how to say that. Anyway, place that I know of, it's amazing. And so we're really lucky to work there. So our study site is a place called Hall's Cave, after the Halls, who own the private ranch that it's on, and they let people go and play with fossils on their site. They've been doing that for about 50 years. And we're also looking at about 50 other cave sites in the area. And this is a picture from inside looking out. This is a 6-foot paleontologist for scale. These are bones. These are bones that washed into the cave just before we were there. So this is a flood deposit. And it's been a pretty continuous flood deposit for 22,000 years. So here is our age model where this is the depth. Here's the column. This is the age. And I've indicated the 1 full glacial, 2 the extinction boundary here. And it's a little light in here, so you probably can't see too well, but you see all these little white things? Those are bones, right? There are tons and tons of bones. And this is a continuous deposition, which means that we can actually get really fine scaled, really fine scaled data. And for some of our analyses, we're doing this at millennial scale for 22,000 years, right? We have over 160 radiocarbon dates, thanks to my colleague, Tom Stafford, who's produced this. Importantly, this site records the presence of an amazing assemblage of mammals, right? So this was like Africa in the Pleistocene. And we had entire guilds of browsers, mixed feeders and grazers. This included things from mastodon and sloths all the way to bunnies and various species of pronghorn. We had camels and Gutherium, and we had mammoth and glyptodont and lots and lots of species of horses. All of these things are present within each of these trophic categories. They're ranked from largest to smallest size. So you can kind of see the size partitioning as well, just like an African savanna. So what happened at the terminal Pleistocene in this location? All of these things went extinct, right? So what was left? A single grazer, the smallest of the 3 bison, a mixed feeder, the peccary, and then a deer and a pronghorn and a couple of bunnies, right? Everything else was lost. Catastrophic change in the herbivore community. In terms of the carnivores, most of which are here, we had an amazing carnivore guild. So we had all of these cats, including the cave lion and the 2, which I think is here, and the 2 species of saber-tooth, as well as jaguar, mountain lions, bobcats. We had dire wolves and so forth, right? And we had Arctodus, the infamous short-faced bear. That's this here, right? Which was supposed to be a voracious predator that chased down and ate horses. What went extinct here were all the large ones. But the extinction wasn't, it hit all the large ones within each guild, particularly hit the Felidae, the cats, because there were a lot of cats in the ecosystem. So in thinking about how this ecosystem would have impacted this site, we think about what a large mammal does, right? So here's our large mammal, and it's walking around the environment, and it's foraging, right? So it's changing vegetation. It's basically a giant fermentation vat. Think of like a beer keg with legs. And so it's actually emitting a lot of greenhouse gases. That's actually something we've looked at, believe it or not. And it's pooping, which changes nutrient deposition and nutrient cycling. So it can lead to changes in greenhouse gases, soil regimes, fire regimes, and so forth if you remove this animal. All of these things, of course, can change the structure and function of ecological communities. And let's not forget what happens to the survivors, right? So the survivors themselves then can respond to the absence of these large body congeners. The things in red are things that we've actually looked at in a bunch of different studies. Here's a few of them. But I'm not going to talk about most of this. So today, what I'm going to talk about actually is just what this community looked like before and after the extinction, and how some of those surviving animals responded. Okay, so I'm going to talk about the ecological niche and how it changed. And I should mention, I'm going to focus today just on animals over a kilo, really bunny size and up. That's mostly because while we're working on rodents, we're not done with them yet. So I can't really talk about them. And this is where a lot of museum work comes into play. So we go museum at this phenomenal collection at Texas Memorial Museum. Here we are museuming. This is the Quonset hut, not air conditioned. Do not go there in the summer that we work at. If you ever been to Austin in the middle of the summer in a metal building, it's really miserable. And here are some of my students working on collecting things. And actually my daughter, who was also working on this project, was at the time. Still is, but. To date, we've looked at about 45,000 fossils, right, passed through the lab. And we've accessioned about 10,000. That's because we take unsorted bulk matrix and we identify fossils and then add them to the collection. That's us returning to the museum some of the trying to be good scientific stewards. And you can just sort of see what they look like. Preservation is phenomenal. So most of these fossils that we're dealing with at Hall's Cave have collagen, for example. So you can do all kinds of things. Okay, so I mentioned body size. So for all of the fossils that we look at body size. I already told you how we do that. The other thing we do is we take advantage of the fact that we have CSI here. The Center for Stable Isotopes, this phenomenal facility that allows us to look at the isotopic composition of bone in this case. And we're looking at bulk carbon and nitrogen values. So how do we do that? Well, we take a sample. We drill a little piece of bone after permission, about the size of a pea. We do some hocus pocus chemical stuff, and then we send it to CSI. That's not very, I didn't explain it very well. It's all right. You know what? It's late. You don't really care. Well, most of you don't care. Some of you. Okay. So let me just, so I said stable isotopes. Here's another thing that I should explain because I'm going to show you a whole bunch of stable isotopes. So let me just try to explain this. How does this work? Hopefully you recall for each element in the periodic table, there exists different species, if you will, right? So here, for example, you have carbon 12 and you have carbon 13. This has an extra neutron, it's heavier. There's also carbon 14, but that's radioactive and we use that for dating, right? So we can measure the actual abundance of these elements in natural ecosystems or in bone and express the ratio of the heavy isotope to the light isotope. And this is relative to a standard, which means the values are in per mil and they're also they're negative numbers, right? So when you see negative numbers, don't be confused. The important part about these different isotopes is that it turns out that biology actually notices that there are different isotopes in the air, and it incorporates these in different levels. So C3 plants tend to favor the lighter isotope C12, and thus have vastly different isotopic signatures than do C4 plants, which favor the heavier isotope. And of course, as you go along the food chain, the carbon isotopic composition of an animal's tissues reflects that of what it ate. So primary consumers end up with different amounts of isotopes in their tissues as well. And you can differentiate a browser from a grazer based on their isotopic signatures, right? So that's sort of how carbon works. Nitrogen is really interesting too. Because we have collagen, we can look at nitrogen. Nitrogen reflects trophic position. So higher nitrogen values mean that something has incorporating increasing protein in its diet. And typically it's about a 3 to 4 per mil shift means a higher trophic level. So carbon is telling you about diet. This is telling you about trophic level. So let me cut to the results. I'm going to show you a bunch of plots like this, where this is carbon, this is nitrogen, and I'm going to show you the species means for now. And you should look at this and ask yourself, where would you expect things to play out, right? So if you're a carnivore, you should be higher. If you're an herbivore, you should be lower. If you're a grazer, you should be over here somewhere. And if you're a browser, you should be over here somewhere. And if you're a carnivore that eats browsers, you should be over here somewhere, right? So visually kind of get that. So what did this community look like? Well, wow, it looked like Africa, really. So we had everybody in here, right? We had a full assemblage of animals from grazers to browsers. To show you some of them, mammoth are here, there's bison here, the various horses are here, mastodon are here, these are deer, and these are camels. And this little 22 here, that's actually the black bear. Turns out the black bear is indistinguishable from any herbivore in this ecosystem. So let me add on to this some of the carnivores. So here is the black bear, here is Arctodus. So you see this particular skull, this giant carnivore that allegedly ate horses and chased them down. It's basically indistinguishable in isotopic space from a skunk, right? It's right here. So that's kind of amazing. In terms of the dogs, here's the dire wolf. And then finally, let's put in what's really interesting. And that's the cats, right? The cats are all super high. This is because they're hypercarnivores, right? So we have bobcats, we have jaguars, the cave lion, and then here are the 2 saber-tooth. And notice how tiny that isotopic space is that suggests it was a specialist on something, right? And I'm going to come back to that in a second. So this is our Pleistocene community, again, carbon, nitrogen. What did this look like then in the Holocene when these things went extinct? Well, it looked like this sad picture up above, right? So you can see there's one grazer left, the smaller bodied bison. This is the jaguar, which shifted way over in isotopic space. But there is plenty of isotopic space that's missing, which is amazing, right? And more on that in a second. So I mentioned that we've done a lot of body size. So how do we incorporate body size with isotopes? Well, we did something I think is a little interesting. We basically did those same biplots of carbon and nitrogen, but now we're going to add contour intervals where we link things that are the same size. And that's done here. So this is nitrogen, this is carbon, and these contour intervals represent animals that are the same bodies, or each contour interval is the same size. What that means is where they stack up and you get cliffs or mountains, that means that multiple animals of different body size are eating or feeding in the same isotopic space, suggesting that that's how they coexist in the system. When we look at the Holocene, what we find is we lost so much of that topographical complexity, right? Many of these cliffs and things are gone, and the overall isotopic space that's occupied by mammals in general is just vastly different. We call that actually the missing pieces. So did these surviving animals respond? Did they shift their diet in after this extinction? And it turns out that some did and some didn't. And today, I think I'm just going to talk about the carnivores because I don't have forever. And I'm sure you probably want to leave at some point. We had this amazing Felid guild. I mentioned that before, everything from like a 300 kilo cave lion, these 200 kilo saber-tooth and scimitar cats, to jaguar that were 100 kilo and mountain lions that were 50 and bobcats that were 10. Beautiful scaling of these things. So how did they coexist? Well, it turns out they coexisted by partitioning their diet. So here, I'm not including the caveman because we don't have as much data to feel confident in it. Here are the other cats. And here is the carbon value of each of these species, right? So this animal is eating grazers, it can't be eating anything else given how small its niche is. And you can see there's sort of a regular progression with size. In the Holocene, this changes, these all go extinct. What we find is that the jaguar, which was here, shifts its diet in carbon space, right? So again, grazers are here, browsers are here. The puma, which was barely present on our site, we only had like 2 fossils in the Pleistocene, becomes abundant and expands its range. And the lynx also changes. Really kind of interesting. And this is also true of nitrogen. So if we look at nitrogen, you can see that these animals are incorporating different nitrogen signatures. And what's really interesting is how elevated these nitrogen levels are for some of these extinct cats, because we wouldn't expect them to be quite that elevated. And it turns out that likely what's happening is that these are cats that are not just eating mammoth, they're eating baby mammoth. And because they eat babies and babies consume their mothers, their little parasites, they basically are elevated above, I didn't mean that towards my children. They basically are elevated 3 to 4 per mil over the adults, right? And this is corroborated by the fact there are hundreds of these little sad baby mammoth teeth, right? That we can find in drawers that have tooth marks on them. And so at some of these sites. Okay, so I showed you kind of those means. What I want to do and I promise I'll be a little more brief. Like, how am I doing on time?
6:32.
Okay, I don't remember when I started, so it's a little hard to say. But anyway, okay, so what I'm going to do is show you the individuals now, the individual fossils and what they look like, right? So this is what it looked like in the pre-extinction, right? So here are the extinct cats. And what I plotted are the individual data for each of the fossils as opposed to the means and the variation. These are what we call Bayesian ellipse areas, which basically, sort of shadow the main area that's incorporated by that. And you can see that these are all very distinct. They occupy distinct areas of this isoscape. After the extinction, and these are the extinct species now, you can see their shifts. And you can see that the Pantheria moved way over here, and the lynx also moved. So you can see that these shifts occurred right after the extinction, which is super interesting. The lynx continue to shift in the modern. So here is the Pleistocene, this is the Holocene, and this is the modern lynx. And what exactly it's eating right now, I suspect it's actually tracking changes we saw in bunnies and hares, but we haven't actually gotten to that level of analysis yet. So let me show you a little bit about dogs, canids, because they do some interesting things. The canids, there were quite a few. They also partition diet with body size, where the largest wolf, the dire wolf, is feeding on grazing animals, and then more towards browsers as you get smaller bodied. And these are highly significant, I should say. In the Holocene, when this goes extinct, there are shifts, and you can see the expansion of some of these niches. There's some really interesting things going on here. This is something we're still working on, trying to flesh out the sample sizes and understand these patterns. And that's also true of nitrogen. So you can see that there is a difference in the amount of protein that was incorporated in the diets, or the amount of flesh they ate, basically. So here's the Pleistocene isoscape, coyotes, wolves, dire wolves. And what you can see is the coyotes don't do much except for expand their range. Wolves shift, and there's some interesting things there too that we hope to look at later. So I'm going to finish by showing you a group that nobody ever thinks about, and that's the poor little mesocarnivores. Paleontologists don't like mesocarnivores. They're boring, right? Apparently neither does the computer. Whoa. Oh, that's weird. What happened here? I'm afraid to touch anything. No, because I'm plugged in, right? Try again. Oh, that's weird. Okay, let's try. I have 100% power, it can't be me. Oh. Whoa. It looks fine on my screen. This seems to be the projector. Okay,
That's me.
okay. Oh,
If we go back to you, it's still.
oh, wow. Okay, so you,
You
Oh,
want-
is that me or you?
This is me, but you can control
Okay,
with
um,
alcohol,
I
like the down.
am so confused right now. So I think he has a PDF of my presentation.
Yes,
Okay.
that is a PDF.
I see. Okay, got it. All right, so let's see what I was. Where was I? Mesocarnivores. Yes, mesocarnivores. Paleontologists hate them because they're boring, right? They don't have big teeth. They're not sexy. Nobody wants to stand up at the Vertebrate Paleo Society meeting and talk about skunks, but they should, because the skunks are cool. So what we find in the Pleistocene is this is what the isoscape looks like of these two different species, the spotted and striped skunk. And notice how they're very different. They're eating different foods. As we go towards the Holocene, they become a little bit more similar. The isoscape expands a little bit. They change in nitrogen and carbon. And then in the Holocene, suddenly they overlap in their isotopic space, which is really interesting. And it suggests that there must be more intense competition. Although that's interesting. We also have a whole bunch of taxa that don't seem to care at all, right? And I feel like there is a meme in here somewhere, given that there's a badger. But I didn't know that meme either. But it turns out that these animals, which don't seem to change over time, maybe they do. Because when we take the individual data and we look at it over time, we find that right at the extinction boundary, there's a huge change in nitrogen and carbon. And we see that for every single one of these mesocarnivores. So something's happening to the ecosystem that they're responding to, but then after that, it kind of resets, and then this is the modern. So again, this is something we're working on, and we'll have to stay tuned. So lastly, I gotta say, most of mammals are rodents. And maybe you don't like rodents, but most mammalogists really like rodents. And so, and with the rodents, it's really amazing because we have thousands of fossils, right? And so we can look at the responses of individual species at the population level each 1,000 years for 22,000 years. And we've been doing that, but we're not that far, we're not done yet. So I'm just going to show you one graph. And here I'm challenging you. I've shown you all these isoscapes, right? So now I'm showing you 15 of them stacked on top of each other. So here's one isoscape, here's another, and so forth. And this is the one for the full glacial. And then as you go up, they get younger, right? And the only thing I really want you to notice is where all the data are. There's crosshairs in these to kind of orient you a little bit. But notice how there's fluctuations in the diet of this animal. This is a Neotoma, a wood rat, over time, right? It turns out that these fluctuations are related to biodiversity loss and ecosystem reorganization, but they're also related to climate change. And that's something we're working on too. And this is where it's really sad that my screen isn't working, because this actually is a movie and I can't show you the movie sadly now, but we got a micro CT scanner last year. And so what we're doing now is trying to incorporate more finer scaled analyses of these rodents. So this is a pocket mouse jaw that I scanned last summer. And again, if you could see it rotate, you would notice that I cut it in half so you could see the roots and the pulp and the enamel and so forth. And this means that what we can do, oh gosh, I'm hitting the wrong button, sorry, is look at a bunch of different parameters of teeth. So things like the enamel crown ratio, which reflects something about the hardness of the diet, or folivory or tooth pointiness and so forth. We can characterize these using our micro CT scanner. And this is something that we hope to do, not just for rodents, which one of my students is working on, and I'll show you in a sec, but also for carnivores, to see whether after these weird changes that occur after the extinction are reflected in their tooth morphology. So the work on rodents continues. So most of these, not all of them, well, okay, only half of them are working on rodents. But here are some of the students that I have right now that are working on Hall's Cave. Marina is doing some really cool stuff with the carnivore allometries. Esteban is going to pick up on the felids, and Carson has done a lot on the larger bodied mammals and also functional diversity measures. Jonathan is sort of our 3D imaging guru. And then because I don't want to leave out my other students, I have to point out that they don't do Hall's Cave, but they do cool things too. So they deserve kudos for that. So what I tried to do today then was point out that the characteristics that predispose mammals to extinction are complicated. And it's not temperature and it's not body size. But that when you see size selective extinctions, that's an indicator, it's a hallmark of human involvement in that extinction, right? And over time, hominids, through their actions, have fundamentally reshaped mammal distributions on Earth and energy flow through ecosystems. Past biodiversity loss has fundamentally changed the way ecological communities work, interactions between species, community structure and function, and the diet and body size of some of these surviving species. And I've showed you some of this today, but there are many other things that we're working on as well. And I would leave you with this thought that these large mammals matter, and their presence or absence in an ecosystem is really important for how that ecosystem functions. And this is an image of white sands in the Pleistocene. So not very far from us, this is what our landscape looked like just 13,000 years ago. And yeah, we had bunnies, but we also had some of the saber-toothed animals and lions and other things too. So let me thank my family for my husband, girls, our very many furry floofs. And I'd also like to thank all the people that work on Hall's Cave. There are a lot of them, both graduate students, postdocs, other colleagues, including Seth Newsom, who was instrumental in the first Hall's Cave grant, and my colleague Kate Lyons, who we just heard that we got a new Hall's Cave grant, which we're really excited about. And we're planning on doing a lot more cool things. And then finally, just my friends, students, and colleagues, of which there are many and varied, and I can't write them all down. So just thank you very much. And again, what an honor to get this, and I'll leave you with this.
Thank you very much, Dr. Smith. I think we do have time for one or two questions from the audience, if anybody has a question that they'd like to ask Dr. Smith.
So you mentioned, the controversy about the dating of humans getting to North America. Is there a way that you get a finer scale for the body mass change to actually get an independent point on that?
So, oh, you mean when we see the shifts in body mass?
Yeah.
Yeah, actually, we can certainly do that for our site. But when they get to our site is a different question of when they get to North America, right? And we actually have charcoal and flint, and there are actually Clovis points and things in this cave as well. So yeah, we totally know exactly when humans got to this cave. That's the charcoal record in particular changes a lot.
Well, I'll ask you a question.
Mhm.
So a lot of the impact of humans was from hunting,
Mhm.
right? But they also did other things like
Mhm.
burn the landscape and
Right.
so forth. And are you getting together a toolkit that allows you to parse out some of those other effects, which then sort of ramify down to affect the mammals, the
Yeah.
other mammals?
Yeah, no, that's a great question. I mean, there've been a lot of studies in Australia, for example, where they've been able to quantify the amount of change that occurred because of fire regimes and other things as well. We have been actually starting to keep better records of the charcoal and things. It might be possible to try to actually look at pinpoint habitat changes that occurred. And there is a vegetation record, a phytolith, and also a pollen record for the site. In the past, we hadn't done much with it. But in this new grant, we're gonna focus exclusively really on the Holocene and try to get at some of these changes that probably occurred. Once people got there, there seems to be intermittent use of the cave over time. I mean, frankly, it's not that great a place to be. It's really dark and it's got lots of pill bugs and it's kind of gross, honestly. But there were probably times when it was a great refuge. So, yeah. So I think that would be interesting and we will look at that more. There was another question. Yeah.
Lawrence Strauss here.
Oh, hi.
How are you? Congratulations.
Thanks.
As a particular question, I often sort of ask in class here, you know, why is it that the horse goes extinct, having been an American
Yep.
species or American taxon, virtually never hunted by humans, very little evidence that humans interacted
Right.
with the horses in the Pleistocene. And yet the bison, which they did end up hunting a lot, was able to survive by downsizing from the giant forms of the late Pleistocene to the small forms of the Holocene. Do you think it's something in the genetics of these two different species?
Yeah.
That both grazers?
Right. No, it's a really interesting question. We're not sure how many species of horses we had, at least 3, perhaps as many as 5. The horse taxonomy is a little confusing, as you probably know. There were 3 species of bison, but one of them might be anagenesis. So the bison, which is the bison we have today, is about half the size of the Pleistocene bison. So it is clear that the only one of those that survived was the smaller form. Some people have argued that was climate change, actually, why it got smaller, but it is also true that there were no longer any really large predators to eat it, not just humans, but other predators in the ecosystem. So it might have been possible to be a smaller bodied mammal and make a living, right? And as to why horses went extinct, all I can say is the evidence is pretty compelling that, we had 16 large-bodied herbivores, 13 of them went extinct. So it wasn't just horses, it was all of them. And certainly it wasn't all by hunting, right? There was some work, actually it was one of Jim's graduate students that I worked with on a project, Wen-Young Su, who was a mathematician, and Rick Charnoff and I a few years, maybe more years than I want to know now. But we worked on a project where we looked at a life history model. And this had been done by John Alrice some years earlier. And we asked the question, did humans actually have to target animals to get them to go extinct? And it turns out that it's actually amazingly easy for a species to go extinct. All humans had to do was add a little bit of extra mortality. And because most species, recruitment and death are sort of balanced, you add a little bit of mortality, they're doomed to extinction, right? If there's no rebound. And that may be why when humans first enter a place, those species that are particularly susceptible, that little mortality does them in, and then some taxa, it doesn't do it. And they persist. Moose persisted, for example, elk persisted. And they're relatively large bodied, but yeah, it's a super interesting question. And I don't really have a good answer for you.
And then when they escaped from the Spaniards,
Mhm.
they were reintroduced.
They did.
and go crazy. And
I know that they do.
There's got to be some change in the competitive environment.
Yeah, it's possible.
thank you, Dr. Smith, for that wonderful talk and presentation. And I'd like to bring Dr. Fisher back up just to close out and also for a presentation of citation.
Yeah, and then I have to say, as a chemist, I love it when biologists use chemistry to do really cool things. So I was like I was like, I know what I know all the elements and all the species of elements. We, in November, when we announced that this year's ARL recipient, we did so during our research and discovery week. And we gave you a great little token then. And so we have a plaque that we're also going to give you, so you have something else to put on your walls besides skulls and things like that. Just really such a pleasure to hear you talk, and I love listening to the decades and decades and eons and, you know, millions and millions of years. When I said the 68th award, that now sounds very pales in comparison to the number of years of this work. So with that, I'm just gonna hand this to you.
Thank you.
And I don't know if we are. Yeah. And with that, there's a reception, there is food and beverage out for everyone. So please join us in congratulating Dr. Smith one more time.